CRISPR is this new technology which is sort of revolutionising the way we go about genetic engineering. It's opened up all these possibilities for genetic engineering that are really now simple and easy because of the way that we've been able to harness this new technology. When I say new technology, the new technology is the way that we use CRISPR, not necessarily CRISPR itself. Because CRISPR is found in bacteria and it's a defense mechanism that they've used in order to try and fight viruses by chopping up bits of the virus's genome. So CRISPR stands for Clustered Regular Interspaced Short Palindromic Repeats. So a bit of a mouthful, hence why we use the acronym CRISPR. So CRISPR contains these segments of DNA, these sequences of DNA, which have this base pairing, which is unique in that it allows them to have this hairpin structure like so, where because of the fact that this sequence is repeating or identical on opposite strands at sort of one end to the other of this repeat, at the end of the day what happens is it's able to have this hairpin structure. In between this hairpin structure we have these segments of DNA called spacers, which might look something like this, before having more hairpins like so. So what these spacers are able to do, so this would be our spacer here, what these spacers are able to do is they are able to latch onto particular sequences of viral DNA and cut the sequences or the segments of viral DNA to try and render the virus unharmful. And these spacers can be added to CRISPR over time as sort of a memory mechanism so that the bacterial cell can remember the segments of virus, what they look like and how to chop them out and defend against the virus in the future. Now, what we are able to do is we've worked out how to harness this CRISPR sort of phenomenon and use it for genetically engineering desired genes of our own. So what we do is we use CRISPR alongside something called a Cas9 protein. In particular, Cas9 stands for CRISPR associated protein 9, and we can use this technology to cut out segments of DNA very, very particularly in order to manipulate DNA. So if I draw a bit of a blob here, this is going to be our Cas9 protein. So this protein can work with CRISPR in order to remove any segment of DNA, any particular gene that we want. So we have this Cas9 protein, and just going to draw in here a segment of DNA that we've opened up. So this is our, I'll just call this our target DNA. So this is the DNA that we want to cut a segment out of, we want to cut a gene out of. So what we also do is we create something called guide RNA or gRNA. So we create guide RNA and it might look something like this. So you can note this hairpin structure. So this is where we see the similarities between CRISPR with this hairpin structure. So what we do is, much like the bacteria has these spaces in CRISPR between the hairpins, which are going to be complementary to a viral section of a virus that we want to cut out, the bacteria wants to cut out to render the virus harmless, we can create a particular sequence as a spacer in our guide RNA or our gRNA, which is going to be very, very similar to that the CRISPR repeat. We can create these desirable spaces of certain sequences that have a complementary sequence to genes of interest on our target DNA. So what this spacer will do is it's going to go along and it's going to bind in a complementary manner to our gene of interest. And then along with the help of the Cas9 protein, we are able to go along and make a cut like so. And then we can snip right through the DNA and remove that segment. So this gives us the ability to remove a segment altogether and replace it with something else if we want to, or just remove a sequence from the genome altogether. So this technology has made it far more easy, far more targeted in being able to locate and remove particular genes from a sequence thanks to this CRISPR-Cas9 protein technology.